Today we're going to discuss literature searching and dissecting a journal article. In the last lecture we discussed research study designs and the different types, and you should have reviewed the statistics that you needed to know. So the importance of a critical research appraisal. So one thing it's important to realize is that just because a study has been published in a peer-reviewed journal doesn't mean it's correct. Approximately 1/2 of published medical journal articles use statistical methods incorrectly and make erroneous conclusions based on those incorrect statistics. So Dr. Eric Rollum, he's a scientist that has an entire website dedicated to detailing how research and statistics have been inappropriately used, and they have current examples of research that has been published and where they've gone wrong. So clinicians and policymakers change their practice based on the results of published research. So this is why it's important to make a critical appraisal, because you don't want to make a change in practice without having critically appraised the study that you're going to be changing your practice to. So as you read an article, you should answer questions about the design, methodology, and analysis before forming a decision. So in order to summarize a study, there's 4 questions that you can use to summarize a research study. The first is, where did the author start? This is found in the introduction and main research question. The second is, what did they do? So these are the methods. What did they find, which is the results, and what does it mean? This is the discussion and the conclusions. So the standard research article is formatted in the following way. There's the title and the abstract, the introduction, the message, which has many parts, including subjects, design, instrumentation, procedures, and data analysis, the results, the discussion, conclusion, and then references, and there may be acknowledgments where basically you thank everyone who helped you. So the research title is usually formatted to state the purpose or the conclusion of the research. It should draw the reader's attention because that is the first thing they're going to read, and it will determine whether they decide to click it and read the abstract. So the title should be brief, approximately 10 to 20 substantive words. So in the following example, which is indeed an article that I published, we can see that there's about 16 substantive words. So things like into and the don't count. So this is summarizing that basically this article investigated the subcellular effects of DHA in a low-density lipoprotein nanoparticle on normal and malignant mouse liver cells. So did DHA kill the normal and cancer cells? Let's find out. All right, so the abstract, after you are intrigued by the article's title, the next thing you would read is the abstract, which is a short 300-word summary of the article, which includes the major aspects of the article, which includes the overall purpose and research problem, the basic study design, major findings, and a brief summary of the conclusions. So readers will read the abstract before they decide if they want to read the entire article, because it takes quite a bit of time to read an entire article. So you don't want to read an article that isn't relevant to what your search topic is. So reading the abstract gives you an idea of whether the article is worth your time or not. So this is an example of an article from, it is my article. This is an abstract. So this one's really nice in the fact that it actually has the different sections segmented by background, methods, results, conclusions, and then general significance. A lot of our abstracts won't have that segmentation. It'll just be one big paragraph. But you can see how like the background's one really long sentence. No, 2 sentences. The other ones, so they're very short, and it's supposed to be very brief. The publishers limit how long they can be. So the introduction should clearly present the conceptual aims of the study. The hypothesis should be clearly stated, but if you have read a lot of journal articles, you know that it's not like they don't usually say the hypothesis is. It's kind of more abstract than that, and they don't necessarily highlight it, or sometimes it's not even clearly stated. Honestly, when you're doing research, you might not know what your hypothesis is. You just want to see what happens. So you're like, I'm going to do this study. I'm not sure which way it's going to go, but I'm going to do it. So you don't necessarily have a hypothesis in mind. And then summarize the existing literature on the subject. So this is kind of the brief background on the topic, but one of the main purposes of the introduction is to convince you why their study is important, which is the rationale for the study. So the methods section includes the subjects of the study. So this is the selection criteria of how they chose the subjects, the sample size, and any potential biases that might have been present in the selection criteria. The study design, so that's things like randomized control trial, how the data was collected, then the instrumentation and reagents, procedures such as treatment groups, concentrations, dosing, testing. And the procedure section is generally detailed enough that you know what they did, but it's not detailed enough that it's a standard operating procedure that you could follow along and do it. And then it also includes the data analysis, which is what statistics were used. So in this one, we can see that cell culture was used, 2 different cell lines were used, they were cultured, and it says like what company the media was supplied in, what the conditions of the incubator were in. And then down here we have that they were cultured in 96 well plates and then treated and then mixed with this reagent to test for viability for 4 hours at 37 degrees and read on a Molecular Devices Thermomax M5 microplate reader. And then we have a little bit of statistics if the relative cell viability was expressed as a percentage of the untreated controls. So kind of this is like a viability assay. We'll see a graph that represents the results of this assay in a second. So the results should present all the relevant data as clearly as possible. There may be additional data in the appendices. Journal articles often limit how many figures and how long an article can be. So if you have more figures, but they're not key figures, you would put them, the authors will put them in an appendix that can be downloaded on the article's website, on the journal's website. And then they should state the statistical test used for each figure. So for this figure, you can see that I think an ANOVA was used for this. And then they also they have the p-values, and the bars represent the standard error of the mean. And if possible, the reader should be allowed to make judgments on whether the author's assumptions for the test were fulfilled. And the test should show the precision of the data. So often these bars will represent the confidence interval, the standard deviation, the standard error. Sometimes there will be little stars showing where statistical significance was reached, and the number of stars will represent different levels, different p-value levels, usually like 1 star is 0.05, 4 stars is less than 0.0001. This one doesn't have stars because there would have been way too many stars. So the discussion clearly answers the questions posed in the introduction. So this explains how the results support the conclusions, and it might also suggest ideas for future research. So the discussion is a place for the reader to ponder questions like, what were the potential sources of bias? Can that bias be quantified? Could confounding bias be a real problem? Is confounding bias anything that wasn't really taken into account of during the initial study design that might affect the conclusions that are being drawn? And are the author's conclusions supported by the results? So the most effective order to read an article is not actually the way in which it is written. So the first thing you want to do is read the abstract to determine, is this an article I want to read? Then you'll read the discussion to kind of get what the summary is, then the introduction, then the results, and then the methods. An expert in the field may read it in the following order, where they read the abstract, look at the results, draw their own conclusions, see what the authors did in terms of methods, then read the discussion and introduction. That's assuming they even bother to read the introduction, because if they're experts in the fields, they already know the introduction material because they've been doing it for so long. So when you're starting a critical appraisal of a journal article, it should include the following 2 things. One, you need to have a structured method by which all key aspects of the research paper are appraised. So you need to have some sort of method that you're using consistently when you read articles to make sure you don't miss something. And then you need to have a balanced and objective review of the strengths and weaknesses of the paper. So things to consider in your appraisal include originality of the study study subjects, what study design was used, is there any bias, were researchers and participants blinded, what statistical procedures were used, and were those appropriate. And then in the following slides, we're going to present some preliminary questions you should address when you begin to analyze an article. So there's 7 questions that you should kind of keep in mind when you're critically appraising a research study. So the first one is, why was the research conducted? So why is it important? What's the rationale? And does it add to the literature on the topic? So this doesn't assume that you would have some idea of what the current literature on the topic is in order to know whether it's added to it or not. Usually they will, in the introduction, explain why their article is amazing and why it adds to the literature. And then, who was the study about? Was the design of the study sensible and appropriate? Was data collected appropriately to answer the hypothesis? Was the bias avoided or minimized? Were preliminary statistical questions dealt with? And does the data support the conclusions? So the first question is, why was the research conducted? So sometimes it's a very similar study design to what other researchers have done in the past, but they've made it larger, they've continued it for longer, or it's otherwise more substantial than previous studies. And then, is the methodology of the study any more rigorous than previous studies? So for example, you might have had a study that did a cohort study or a retrospective study, and this new article is doing a case control, or is doing like a randomized control trial. So something that's more rigorous. And then, will the numerical results of the study add significantly to a meta-analysis of the previous studies? So if there's debate on a topic, this can help kind of tip the scales towards one side or the other. And is the population that was studied different in any way from previous studies? So for example, previous studies could have looked at a predominantly white male population, and this new study is looking at a much more diverse population. So the second question you should ask is, who is the study about? So are the results applicable to your practice? So if the article was about people in the Middle East and a disease that's common there, then that might not be relevant to your practice in Utah. How are the subjects recruited? So for example, if you are doing a questionnaire on the usefulness of the public library and you're standing outside of the library asking people these questions, then they're biased towards thinking, yes, there's usefulness to the public library because they're already at the library. They already found a reason to go there. Whereas you would be better off surveying the community at large. You also want to look at who was included or excluded in the study and what the criteria were, because having clear inclusion and exclusion criteria can create cleaner results, but you may miss certain aspects of the population, and the results may not be completely generalizable to the population. And then studies outside of real life. So if you're doing a study, was it in real life circumstances or not? So studies outside of real life can cast doubt on the applicability of the results to the general population, but it also does minimize some confounding biases as well, because you can't control real life 100%. Real life is messy. So there's pros and cons to doing real life circumstances. And it's important to know that the sampling and selection criteria can skew your results, and you should be aware of that. So the third question is, was the design of the study sensible and appropriate? So the terminology used in study designs can be intimidating and confusing. So in order to kind of pare it down, two questions you should ask is, what specific interventions or other maneuvering was being considered, and what was it being compared with? So what's the independent variable and what are the controls? And then what outcome was measured and how was it measured? So what's the dependent variable and how are they measuring those outcomes? So the fourth question is, was data collected appropriately to answer the hypothesis? So some data measurements are more subjective and prone to error. So if possible, those should be avoided, just because subjective data isn't as rigorous as objective data. So some of these subjective measurements include symptomatic effects like pain. So pain is very subjective. So some people have a very high pain tolerance and some people might have a very low pain tolerance. So even though they're experiencing, quote, the same pain, one that might think it's a 10 and one might think it's a 2. So there's also functional effects like mobility, psychological effects like anxiety, or social effects like inconvenience. And this has to do with like quality of life. So you can use scales to help quantify some of these effects by using like a pain scale of like, on a scale of 1 to 10, how would you rate your pain? But once again, that's still subjective, but it does give you some kind of quantifiable measurement. And then if possible, these subjective measurements should be replaced or supplemented with objective measurements like lab values, mortality, things of that nature. So the fifth question you should ask as you're appraising an article, was bias avoided or minimized? So systematic bias is anything that erroneously influences the conclusions about groups and distorts comparisons. So basically anything that kind of muddies the water. So whether a study is a randomized control trial, a non-randomized control trial, cohort, or a case control study, in an ideal world, the two groups should be as similar as possible, except for the difference being examined. And the group should receive the same information and assessment. So basically, you want these two groups to be as similar as possible, except for what you're actually studying. So the assessment should be blinded to minimize bias. So if the researchers know who's getting the treatment and who's getting the placebo, if a patient that was assigned to the treatment group gets a weird result, that researcher is probably more likely to recheck that value. Whereas if that person was receiving the placebo, they might not recheck it. And this creates a performance bias. So systematic bias can be avoided, but depending on the study design, there's different ways you avoid it. So in a randomized control trial, you select, there's very minimal systematic bias in a randomized control trial because you're basically choosing your population and then you randomly assign them. And since it's random, there's not really a chance for systematic bias to be in place because it would affect both groups equally because it's random who gets assigned to what group. So in a non-randomized controlled trial, you kind of use, there's not really a lot of statistical methods to determine if there's bias. So you kind of do a common sense analysis to determine if there's minimal baseline differences in the groups once they're divided. And then in cohort studies, you control for systematic bias during the data analysis portion with some statistical adjustment for differences. So this is like when you're reading a research article and they're like, after correcting for smoking history and diabetes, we found this. So that would be kind of how they control for those sort of biases. And then in a case control study, this is the one that's most open to bias and it's most open at the beginning of the study when you're determining when a person became a case and who was a case versus who was a control. And it's not so much a bias of determining the outcome, but rather who's a case. So the sixth question is, were preliminary statistical questions dealt with? So these answers are usually found in the methods, and these include sample size, follow-up duration, and follow-up completeness. So the sample size should be big enough to detect a statistically significant effect or conclude that there's no effect. So the null hypothesis is correct. But this has to do with the power of the study. So there are statisticians who are fantastic at this, and they can tell researchers, okay, for this type of study, this is how big your sample size needs to be in order to ensure that you see a statistical effect. You can see the difference. So if you're doing a study with humans, the IRB will require you to figure out the subject sample size that you need in order to get statistical difference, but without being like huge. And then the follow-up duration should be long enough to guarantee that an effect can be seen, and it should be complete enough to avoid non-response bias due to losing subjects. So with follow-up, you're kind of treading a delicate balance between you want it to be long enough to see an effect, because if you're studying something like asbestos on lung cancer formation, if you look at the people who have been exposed to asbestos and those who haven't, and you follow them up for a year, you're probably not going to get very many people with cancer. But if you follow them up for 15 years, you're probably going to see a lot more cancer cases. But 15 years is a really long time, and you might lose a lot of people to follow up. So that means, like, when you lose someone to follow up, that means they're lost. You don't know what happened to them. They disappeared. So you can't contact them anymore. They decide to pull out of the study, things like that. So you want it to be long enough that you see the effect, but not so long that you're losing half your subjects. And then the last question is, does the data support the conclusions? So you want to look at the results and then draw your own conclusions based on the results. And then you want to look at the conclusions of the author and see if your conclusions match theirs. In a perfect world, they should. The author might have more knowledge on the subject than you. That influences their conclusions. And you also want to make sure that the author hasn't made any assumptions that they shouldn't have. So after doing this critical appraisal, this is what any journal article reviewer does when they're trying to decide if an article should be published. So possible reasons why papers are rejected for publication. One reason is that the study did not address an important scientific issue or was unoriginal. So they're not going to waste journal space on a study that doesn't matter or has been done 100 times. A study did not test the stated hypothesis. A lot of times authors will circumvent this when a paper has been rejected by rewriting the hypothesis so that it reflects their study, and then resubmitting it. A different method should have been used. And usually a reviewer will call them out on that, and then the authors have the opportunity to complete that study and resubmit. Sample size was too small, so it needs to be larger. Study was not controlled at the appropriate level. Statistical analysis was incorrect and inappropriate. This could be solved by reanalyzing the data, drawing conclusions based on the new analysis, and then resubmitting. Unjustified conclusions were drawn. There was a conflict of interest. A conflict of interest is required to be disclosed in the article. And a conflict of interest is when one or more authors could benefit financially from the publication. So for example, say one of the authors is a major shareholder of a drug company, and the article is about that drug and how it is the magical cure to all cancer. That author very much has a conflict of interest. So any study they do might be inherently biased towards thinking that the drug that they're going to profit from works. And then it might just be a really poorly written paper. I have reviewed papers and read papers that are just horrible for grammar and understanding and just need to be completely rewritten. So now we're going to talk about an advanced literature search and how to do one for your continuing education project. All right, so the purpose of an advanced literature search. So first of all, an advanced literature search is a considered and organized search designed to find key literature on a topic. So it helps you find the current literature on a topic. So this is, so you can utilize the information in your professional practice. You can avoid repeating research that's already completed, and it can help you formulate and plan your own original research study. So you can say you have a study where you're trying to look at the effect of oxidative stress on lysosomes using your drug of interest, and you look at other studies that have looked at oxidative stress on lysosomes using different drugs. You can look at their methods and figure out how did they test to see if there was oxidative stress. So you can kind of steal methods. Also, you can use this information to kind of design your own study. So a literature search is different from a literature review, which is different from a systematic review. So a literature search is the considered and organized search to find key literature on a topic, and you create a summary for your own personal use. So this helps you get information on a topic. So the first thing you do is you define your search topic. You select your search resources. So these are like your search engines and databases. You choose your search terms, which is what you're going to plug into the search engines. And then you compile your search strategy and execute your search. And then you review your results, read your articles, and organize the results and draw your conclusions. A literature review, on the other hand, goes a step further than a literature search, and this is usually conducted by one or two scientists, and it looks at a large amount of the literature on a topic, qualitatively summarizes it, and analyzes the search findings on a topic. So it searches several databases on the topic, critiques some articles. There may be biased articles in there, and then it looks at those articles and informs the audience about the topic. And this is usually, this review is usually published in a journal, in a peer-reviewed journal for other people to read. So a systematic review goes even one step further than a literature review, and this is a high-level overview of primary research that identifies, selects, synthesizes, and appraises all high-quality research. Basically, it looks at every article very closely and is used to guide best practice. So this is a systematic search of all databases. It has clear inclusion and exclusion criteria of articles that will be in the systematic review. It critically analyzes all the relevant articles. It eliminates any biased articles, which is something that a literature review doesn't necessarily have to do. And this is usually done by many authors that are experts in the field, and they collaborate for months to years because it takes so long to do it. And a systematic review may include a meta-analysis of the primary data, but it doesn't have to. And this is used to guide best practice. So this is like, we have looked at everything, we have compared the different things, the different conclusions of all these articles, and this is what we think is best practice. So a literature review is qualitative and it's a summary of the literature. A systematic review is more quantitative and is designed to answer a specific clinical question, and it may include a systematic review, but it doesn't have to. And the meta-analysis is a systematic review that analyzes the primary data and draws its own conclusions. So now we're going to talk about how to perform an advanced literature search. So the first thing we're going to do is define our search topic. So I just chose one of the continuing education topics from the Excel sheet, and this one is current and emerging laboratory testing methods for screening, diagnosis, and prognosis in Alzheimer's disease. So the first thing I'm going to do is figure out the keywords in my topic. So I think the main keyword here is Alzheimer's because I don't really care about any of these other things for other diseases. So I want Alzheimer's in there, laboratory test, screening, diagnosis, and prognosis. Probably have to search for each of these separately. And then assay, biomarker, because assays test biomarkers. So this one isn't in there, but it's relevant. So this would kind of be what my keywords would be for a search topic that I've come up with. And then I would look at my search engines and databases. So one good search engine is Google Scholar. This is different than just regular Google. You have to go to Google Scholar. So that's scholar.google.com. And it pulls up peer-reviewed journal articles. There's also PubMed, which is kind of a repository of all articles, but it often doesn't have the full text article directly in there, and it will link out to a full text article. Scopus, Web of Science, Medline, ResearchGate, and then you can also do a search on the university library. They have their own set of databases, and you can also do just a general search that will search all of the university's available databases. And the best way to get access to full text articles is if you access it while you're on campus or using the VPN or log in to the university library's website. All right, so some tips and tricks to keyword searches. First, you identify your keywords, which we did 2 slides ago, and then you want to figure out how to narrow or broaden your search. So first thing to look at is synonyms. So assay is the same as test, analyte is the same as biomarker, so you'd want to include, do searches with either of those. And then you can also use exact phrases where anything that's within quotation marks will be searched as an exact phrase. So this would search Alzheimer's disease screening exactly as it's written. Capitalization doesn't matter. You can also use Boolean logic terms. So these and, or, and not, when they're in all caps in a search, will cue the search engine that it has to include or exclude certain key terms on either side of those Boolean logic terms. So for and, it means you have to include both search terms on either side of the Boolean logic. Or it can be either search term, so this would be like assay or test you could have on there. And then not, this is useful when you're getting a lot of irrelevant articles. So like for ours, if you're looking at Alzheimer's, you might be getting a lot about dementia, and you don't want to hear about dementia. So you could put Alzheimer's not dementia on there. And then you can also use truncated search terms. So this little star, the symbol might vary based on the search engine that you're using or database. There's an index usually available that will tell you what symbols mean what. So for this, the star means that anything, it's kind of like just truncated. So it will pull up diagnosis, diagnostic, diagnostician. So anything after that S is kind of, it doesn't matter, just as long as it has the diagnose part. And then wildcard search terms, a question mark is a common symbol for this. This is useful especially for English versus American spellings of things like behavior with or without the U, or looking for various pluralizations of things like woman versus women. And then you can also do an adjacency search. So this doesn't work on all search engines or databases, but when it does, it's very helpful. So the ADJ stands for adjacency, and then the number says how close you want the 2 terms to be, so Alzheimer's and assay. So the closer words are in an article, the more likely they are to be related. So for this Alzheimer's ADJ3 assay, we're looking for articles that have Alzheimer's and assay within 3 major words of each other. So they're most likely related. This will help narrow your search. You can also do subject heading searches. So some databases have controlled vocabulary terms or subject headings to describe what an article is about. So if you find a relevant article, you can see what subject heading that database classified it under and then search for that subject heading. And you might end up with including relevant articles that don't contain your keywords but are still relevant. You can also do citation searching. So say you find an article that you like, you can look at what they cited. And there might be some good articles in their references. You can also limit the date range to keep the search to the most recent articles. So for our continuing education project, I want articles within the past 3 to 5 years. You can also limit by language. So I was doing a search the other day and I pulled up an article that seemed really awesome and I didn't realize it was in French and I do not speak French. So I couldn't read it. So sometimes this is very relevant to help you because you cannot read an article you cannot read. This is also, if you're doing a topic, you might get a lot of Chinese articles as well. And then you can also use search limits or filters. So a lot of databases allow you to limit your searches by publication year, language, publication type, and you want to apply these at the end of your search because you don't want to narrow your field too quickly. You want to kind of narrow your field after you know if you're going to get a lot of search hits or not. And then search filters are pre-tested strategies that identify specific literature. So these filters can include limiting your results to randomized controlled trials, cohort studies, or observational studies. And some databases have these types of search filters and others do not. And then how to keep track of it all, because there's a lot involved in doing a literature search. So it can be difficult to remember where you have searched and with what search terms. So it's nice to keep an activity log, and I have included a template for you on Canvas. And you can also save your articles to a reference manager and then save your searches on databases. A lot of databases have that option to save your search. And you can also sign up for emails on some databases where it will email you if there's any new articles that match your search criteria, so you stay on top of the subject. All right, so this is our template of our search activity log. So you have your research question, which is your topic, and then where you're going to search would be like Google Scholar, Medline, PubMed, that type of stuff, or the university library. And I am going to walk you through how to do a literature search on the university library website, and then what keywords you're using, and then also your parameters that you put in, like within 2 years, English language, peer reviewed, full text, articles only, and then the total number of search results that you pull up. All right, so we are going to do a demonstration on our little Alzheimer's disease topic. So these are the keywords that we've come up with, and I am now going to go to the internet. All right, we are at the internet. So I'm going to put in the University of Utah Library website. Go to the Marriott Library. You want to make sure that you're logged in so that you can access full text articles. And you're like, hey, I don't have any loans, woohoo. All right, so we're going to go to new search up here, and then we put in our search criteria. So I'm going to do Alzheimer's. Biomarker prognosis assay. Okay. And then it has its default settings of books, articles, more, all resources. And I've got 7,000 hits. That's a lot of hits. So I've got some books. Ooh, I've got a novel prognostic biomarker for preclinical Alzheimer's disease. This sounds amazing. I like that one. All right. So this is pulling up everything, and we can see here that we have peer-reviewed journals, we have full text articles, open access. We can decide if we want what type of resources we want, the locations, years. I don't want 1997. So I am going to set this to 2015. And then I'm also going to set it to English language because I can't read any other language. And I want peer-reviewed journals, and I want access to the full text. I'm going to apply those filters. All right, I'm now at 3,420 articles. Woohoo. All right, so let's see. Let's read some synaptic proteins in CSF as a potential novel biomarkers for prognosis in prodromal Alzheimer's disease. That sounds fun. Let's find out what happens if we click it. All right, so on the University Library, it says it's peer-reviewed, it's open access, the full text is available. So this is where we can try Mendeley. So on Chrome, you'll go over here and click the Mendeley Web Importer. And if you haven't used it before, it will ask you to sign into your account. I'm going to choose that. And I'm going to add it to my library. So I'm going to add it. And then if we're lucky, it will add the PDF and the citation in one go. If it doesn't, you can follow this link and download the PDF and then add it manually to Mendeley. But we were lucky, it did download our PDF. So woohoo. And then you could go back and add another one. So let's try this one. And, right, no, it's not showing the right things. Awkward. There we go. Okay. All right, so I was able to do the PDFs. Now we're going to open Mendeley. And going to sync our library so it pulls it up. All right, so these are the articles we just did. Oh, lies. Oh, okay, it's downloading the PDF. So sometimes it takes a second. And right now they're showing up in my library, but not in the group. So you can, the green dot indicates that you've read it or not. And then you can drag and drop it into your group. So now it's in your group. And what's nice is that when you're reading it, you double click it to open the PDF. You can highlight. Sometimes on PDFs it gets a little weird about where it highlights. And you can also use note features. And with the notes, interesting background. With the notes, it shows up over here, and then you can also see them as well. So this is what you would do, and then you can see, you can mark it as read or not read. And that is how you do a literature search. So you would add in your articles, you would give the abstracts a read, and then do a critical appraisal and determine which ones you want to include in your continuing education project. So go forth and literature search, please.